Good afternoon, ladies and gentlemen. I'm Answer Estreisen. I will be presenting the formal test for the grade 12 that was written on the 20th of April today. So let's start. The formal test is test 2.1, and let's look at the memo for that. It is important in every test to ensure that all the instructions is followed, and just emphasize your learners to start each question on a new page. It helps them to prepare them for the end of the year exam. So just make sure that all the instructions are followed, that they answer all the questions, and so the monomers of nucleic acid are shown as, and the correct answer will then be nucleotides number B. That would be the correct answer. Number 1.1.2, nitrogenous bases in DNA always combine as follows: cytosine with thymine, thiamine and adenine, guanine and adenine, and thiamine and guanine, and then the correct answer will therefore also be the B option there. Thiamine will connect to adenine. That would be the correct answer. Number 1.1.3, answer the question that follows the diagram below. Which is the correct sequence of the structures in the male reproductive system that sperm move through during ejaculation? So if we start here, the correct answer will then be, we will have to start here at G, moving up through the vas deferens, moving through down to the urethra. So the correct answer will then be where we have structure G, that would be the semen tubes, structure F, the vas deferens, and then structure C, that would be the urethra there. So the correct answer in this case will then also be number C. That would be the correct sequence of the structures in the male reproductive system that the sperm moves through during ejaculation. If we go to the next question, this would be the section on 1.2. Give the correct biological term for each of the following descriptions. Write down the term next to the question number in the answering book. A nitrogenous base present only in a single-stranded nucleic acid, and that would be uracil. That is the only correct answer there. Uracil. The phenomenon where chromosomes pairs do not separate during meiosis, that would then be non-disjunction. The gland which supplies the main source of nutrients of a sperm cell will then be the seminal vesicles. Vesicle. The place where in a mammalian embryo normally develops, that would then be the uterus. So if we look at the memo that we have here up until this stage, that is the correct answers according to that. If we go to the next part then, question 1.1.3, indicate whether each of the statements in column 1 applies only to A, B only, both A and B, or none of the items in column 2. Write A only, B only, both A and B, or none, next to the correct question number. So the first one there is the scientist who took X-rays that led to the discovery of the double helix structure of the DNA. So the emphasis there is on the X-rays. So nor Watson or Crick were involved in that. It was Rosalind Franklin, and she is not mentioned here, so the correct answer will then be none. None of the above will be correct. To 1.3.2, the male part of a flowering plant where meiosis occur. So the first one, the oval, is in the female part of the plant, and the anther will then be the male part of the plant, leading us to that B only will be the correct answer because A is from the female part of the plant. 1.3.3, the site of normal fertilization. The normal fertilization will then be in fallopian tubes. The vagina only receives the sperm during copulation. So number A, it will also be A only. That would be the correct answer according to this. So according to the memo, there's the memo up until now, there's no mistakes or there's no corrections there. The memo is 100% correct. So let's continue then to the next part. That would be question 1.4. Read the following extract and answer the questions. Few fathers go to greater lengths to protect their young than the seahorse. Uniquely among vertebrate, seahorses nurture their eggs and embryos within a male broad pouch. A female seahorse lays several thousand eggs into the male pouch where he fertilizes them with sperm deposited directly into the broad pouch. After fertilization, he then protects the embryos until they hatch. The lining of the pouch secretes a nourishing fluid that feeds the development of the young. After about 2 weeks, contractions of the pouch expels the young seahorses. Shortly after birth, the young seahorses are able to swim around freely. So this question already has a discussion. So question 1.4.1, according to the extract, is the seahorse oviviparous or ovoviviparous? So according to the memo, the correct answer will be oviviparous. So the next question will then be state one reason for that specific answer you have there. So if we go to the memo states clearly that eggs develop in a pouch and the young are born alive. So we are focusing here on the fact that they say they are born alive and they are kept inside. They are kept inside the male's body, and usually when we say it is oviviparous, it is supposed to be the female's body, but because it is kept in a body, then we are still going with oviviparous at this stage of the discussion. So the next question will then be 1.4.3. Use information from the extract to provide one biological term for each of the following. After fertilization, he then protects the embryos until they hatch. So protects the embryos until they hatch would lead to parental care. So there we will have parental care as correct. And the next question will have shortly after birth, the young seahorse are able to swim around freely, meaning that it can look after itself, and that would be precocial development according to the marking guidelines. So that brings us to the end of section A. Section A is 20 marks allocated in this paper. continuing to the next part, coming to section B, we all know that we have to practice now because there's no section C anymore in any papers for the grade 12s, 10s and 11s. So we need to emphasize this and we need to practice this. So the first question we have here is the diagram represents a phase of meiosis. So if we look at that, we already know we are looking at meiosis. So we have two different chromosomes in the cell, and then we are looking at the next questions. Identify the label number A. The label number A will then be the cell membrane. Cell membrane. It is important to emphasize this with your learners, that the nucleus membrane has disintegrated in prophase already. So the only membrane that is left here is the cell membrane, because the nucleus membrane was as disintegrated when we started with prophase 1. So the next label we need to identify will then be label number B. Label number B will then be the centriole. Centriole or centrosome. Either one of those two will then be correct for label number B. Which phase of meiosis is shown by the diagram? And give one reason for your answer. So if we look at the diagram, all of the chromosomes is arranged at the equator, and therefore if we look at the memo, the correct answer will then be metaphase 2, and the reason is it is the chromosomes are arranged on the equator, but the reason for being metaphase 2 is chromosomes position themselves in a single row. I think we can better do that by saying single chromosomes. Single chromosomes position themselves on the equator. So it is important to identify the fact that it is a single chromosome. It is no more homologous pairs that is involved in this action. So it is important that it's the emphasis is on the fact that it is a single chromosome that positions itself on the equator. Then if we go to the next question, is this a representation of a plant or animal cell, and give a reason for your answer. So if we go back to this cell, we have to identify this as an animal or a plant cell. So in this case, there's only a cell membrane there. So if we look at the memo, we will say that it's an animal cell. That is, as you can see, a compulsory mark. So we mark it with an asterisk like that. And it has only one cell membrane that is visible, or you can allocate a mark for there's no cell wall according to this. So one mark can be allocated for either one of those marks. And then a next option will be there is centrioles or centrosomes. It is present. It has centrioles or it has centrosomes. That is a characteristic of animal cells. So that is also a reason why that is an animal cell. So there is a maximum of 2 marks. Just make sure there's one reason, and the compulsory mark is then for the animal cell in that part of the question. If we go to the next one, name the process responsible for the appearance of chromosomes at region X and give the purpose of this process. So I think it's quite clear here. The difference here at X is the fact that crossing over occurred, and if we look at the memo, we can look at the fact that it is supposed to say crossing over, and it is very important for genetic material to exchange. If we look again at the question, name the process responsible for the appearance, that would be crossing over of the chromosomes at the region, and give the purpose of this process. So again, the purpose will then be to exchange genetic material. That would be the end then of question 2.1, and that is 8 marks in total if we look at that. If we turn the page, we go to the next part, question 2.2. The diagram below shows several events that occur in the human female body during reproduction. So the first part is identify structure A. So if we have a look at this diagram, we have the ovary there, we have the different follicles in the ovary, and then we have the fallopian tube with the different stages of the development of the zygote. So after fertilization has taken place, we have this different shapes of the development of the zygote will develop further in the different stages before implantation will take place into the endometrium wall. So the first one is structure A. We need to give the label of structure A. That will then be the Gravian follicle. Gravian follicle. That would be structure A. The process occurring at number C. So if we look carefully at number C, we are used to what are happening here, but let's look carefully. There's the nucleus of the ovum, and there's the sperm, and there's a lot of sperm out there. So the process that is occurring there, the process will be then fertilization. Fertilization. Number C, the stage of embryo development, represented by number D. So we all know we have the ovum. It will be fertilized by the sperm. We will have the zygote. The zygote will develop more through meiosis. Oh, now I'm lying. Through mitosis. And then we will get to this structure here. And that structure we will identify as the morula. That would be number D. The morula's characteristic is the fact that it is a ball of cells. So there's a lot of cells, there's no space in there. So that is how you can identify the morula. It is a lot of cells being dividing in there, and there's no spaces in between. Then further development shows that there's a big space there that is filled with fluid, but if you can see the big space there, you can identify that as the blastocyst. So it is just important to make sure that your children are able to distinguish between the morula and, in this case, the blastocyst. But they ask number D, and then that would definitely be, according to our marking guidelines, that would then be the morula. So we have the Gravian follicle, fertilization, and the morula. If we go to the next part, 2.2.3, describe the negative feedback mechanism between FSH and the hormone named in question 2.2.2. Okay, so let's first go back to 2.2.2 because I skipped that now. Name the hormone produced by structure B. So here we have the follicles that developed. So primary and developed. So the very important one there is the Gravian follicle. The ovulation took place and the ovum was discarded through ovulation. And there we have the corpus luteum. So if we look there, the correct answer will be the hormone that is being produced by structure B will then be progesterone. The hormone that will be secreted by structure B, if we look at the memo, will then be progesterone. Going back to the question on the negative feedback between FSH and then the hormone in question 2.2, that would then be progesterone. So the negative feedback mechanism between those two, if we look at the memo, there's high levels of progesterone inhibits the release of FSH. So the higher the levels of progesterone, no FSH, or it inhibits the release, it will be very low, which prevent the development of any other follicles. So no other follicles will be formed during this situation. So while the progesterone is high, no other follicles will be secreted and the FSH will be suppressed. It will be inhibit the secretion of that. Name one consequence if the level of the hormone named in question 2.2 decreased in a woman who is 7 weeks pregnant. So if she's 7 weeks pregnant, we can have a look here, then implantation has already taken place. So 7 weeks pregnant, we will then go to the memo, and that would lead to a miscarriage. Or any explanation in that regard, saying that the uterus wall or the endometrium wall was shedded, and with that the embryo that is developing, that would be a perfect explanation of it, and that is correct also. So that brings us to the end of question number 2, and that is 15 marks. Question number 2. So if we go to the next question we have here, that is question 3. The diagram below represents a part of the process of protein synthesis. So we have here, you can see clearly the nucleus pores, where through the mRNA will move to the ribosome. But they are saying it's protein synthesis, so that is making it easy for us. So this double structure here will then be the DNA, because it is unwinded so that we can duplicate, or transcription can occur. So now they are saying, name the process that is taking place in the diagram. So as I said now, the correct answer will be transcription. I will go back to the memo now. Where in a cell does this process occur? So it is inside the nucleus. That is important. And then we go to question number 3.1.3. Tabulate two differences between the process named in question 3.1.1 and DNA replication. So that would then be transcription. And we will compare that with DNA replication. So according to our memo, let's look at that table we have there. So again, 1 mark will then be allocated, 1 mark for the table. So the 5 marks we have, 1 mark will be allocated for the table. Just write a T there somewhere so that we know you allocated a mark for the table. And then we need to mention 2 differences between transcription and DNA replication. So during transcription, mRNA is formed, and during DNA replication, 2 identical DNA molecules are formed. That would be the first mark, the first difference that we will discuss, and there is 1 mark for transcription and 1 mark for DNA replication. Then the next one will be the product that moves from the nucleus to the cytoplasm. So in this case it will be the mRNA moves from the nucleus to the cytoplasm. The DNA replication, the product, meaning the replicated chromosome, will remain in the nucleus. So that is the first 2 options that we have there. But I would like to add on to that, because I think there's also other differences that we can allocate marks for. Transcription will use RNA nucleotides, and DNA replication will use DNA nucleotides. Transcription will use only 1 strand as a template. DNA will use both strands as templates. In forming the mRNA, adenine will be binding with uracil. In DNA replication, adenine will be binding with thiamine. So I think those are 3 very important differences that we must add there. In any case, I think I will start with those differences in teaching this important part of the difference between transcription and DNA replication. So if we go back there, just make sure it's 5 marks and just add on the differences. In transcription, we have RNA nucleotides. We have 1 strand that is a template, and adenine is combined with uracil. And during DNA replication, we have DNA nucleotides. Both strands are used as templates, and adenine is combined with thiamine. So that is important differences that you can add there when marking the question on the differences between transcription and DNA replication. Moving on to the last question. Let's see. An investigation was conducted on the concentration of hormones of 30 women which age between 20 and 30. The investigation was done over a period of 28 days. All the women were put on the same diet. The instrument used to measure the concentration of hormones was the same in all women. The measurements were taken by the same person, and measurements were taken at the same time of the day. The table below shows the results of the investigation. So there we have 0 days up until 28 days. We have the average hormone concentration, arbitrary units for progesterone and estrogen. That are the 2 hormones that we are looking at in this investigation. What evidence from the table indicates that ovulation occurred around day 15? So if we look at that, on the memo we can see that there is a definite increase, allocate a mark for the fact that they know it's increased levels of estrogen. Because this investigation is only done on progesterone and estrogen, this is the only information we are allowed to use. And as you can see, there's a significant increase in the estrogen concentration. At this stage, I would just like to emphasize to the Afrikaans and refer to the Afrikaans memo. If we look at the Afrikaans memo, it says 'n verhoogde maximum vlak van progesteroon. So please just make sure that that is not correct. It is supposed to be estrogen. So on the Afrikaans memo, there is a mistake there. The maximum vlak van estrogen. So just make sure if we are marking that. So what evidence do we have? The increased level of the estrogen. Then number 3.2.2, explain the decrease in progesterone concentration between day 25 and 28. Now again, I think when marking the scripts of 2020, the exams, I think there's quite a few options that were asked where we had discussions that we normally don't have, because we had a lot of questions that was asked in a backway situation that wasn't asked like that before. For that reason, and exactly for that reason, I would like you to look at this question and explain the decrease in progesterone concentration between day 25 and day 28. So if we look at the memo, it is important to remember that the decrease in progesterone is in the question. So fertilization did not occur. We all agree that would be correct. Fertilization and implantation did not occur. Therefore, the corpus luteum starts to degenerate. So again, those are the two marks allocated here. So I also think it is very important that you mention that the corpus luteum secretes the progesterone. Therefore, it decreases. So I would really like for us to also add that the fact that the corpus luteum will secrete progesterone. And because the corpus luteum is degenerating, there will be less progesterone. We won't allocate a mark for a decrease in progesterone. We won't allocate a mark for less progesterone. But it is of the utmost importance that they can say that the corpus luteum is degenerating and therefore secretes less. Well, because it secretes the progesterone and the corpus luteum decreases, therefore the progesterone levels are now low. So again, let's emphasize the first mark will be allocated for the fact that we said that fertilization did not occur. And then the corpus luteum starts to degenerate. And why is that important? Because the corpus luteum is responsible for the secretion of progesterone. And if the corpus luteum degenerate, then there will be less progesterone being secreted, leading to a decrease in the progesterone levels. Number 3.2.3, why did the investigator use 30 women rather than 5? Again, it is important that we look at validity and reliability every time that we look at the investigation. So the correct answer here would be, he used a big sample size, so he improved the reliability of this investigation. he increased the reliability of this investigation, allocating a mark for that there. The next question will then be state the following for this investigation. 2 factors that were kept constant. 2 factors that were kept constant. So if we look at the question again up here, they said all the women were put on the same diet. That would be correct. The instruments that are used to measure the concentration of the hormones are the same. The measurements that were taken by the same person is important, and the measurements were taken at the same time of the day. Those are the things that need to be kept constant. So if we go back to the memo, we have those different options there, 4 of them. The same diet, the instruments, the person, and the time of the day will be kept the same. So any 2 of them will then be correct. I also think it's important to just mention the concentration of hormones of 30 women, and they were between the ages of 20 to 30. So they selected or women between 20 and 30. So these are the factors that needed to be kept constant. So if we go to the last question on this, what is the dependent variable in this investigation? In this investigation, on these different days, we measured the amount of progesterone and estrogen. So the correct answer there will be the hormone concentrations will be the correct answer. So that is only 1 mark. Ladies and gentlemen, this brings us to the end of this. There's 50 marks for section B, and with the 20 marks of section A, that would leave us with a total of 50 marks for the test, formal test 1.2 for the grade 12s. Thank you very much.